Good day everyone. We are the group 3 and we will tackle about microbial genetics. At the end of our discussion, you can able to explain how DNA serve as genetic information, describe the process of replication, transcription, and translation, and identify the importance of microbial genetics. Microbial genetics is a branch of genetics concerned with the transmission of hereditary characters in microorganisms. Microorganisms include prokaryotes like bacteria, eukaryotes like fungi and viruses. Microbial genetics has played a unique role in developing the fields of molecular and cell biology, and also has found applications in medicine, agriculture, and the food and pharmaceutical industries. Microbial genetics also leads to increase the yield of antibiotics from other microbes. Central dogma explains the flow of genetic information from DNA to RNA to make a functional product, which is a protein, and DNA contains the information needed to make all of our proteins, and that RNA is a messenger that carries this information to the ribosomes. The central dogma of molecular biology, coined by Francis Crick in 1958. Deoxyribonucleic acid, or simply DNA, is a long molecule that contains our unique genetic code. Like a recipe book, it holds the instruction from making all the proteins in our bodies. DNA also has a unique double helix shape, like a twisted ladder. It also has a strand composed of long sequence of 4 bases, which is A stands for adenine, C for cytosine, G for guanine, and T for thymine. The bases on one strand of the DNA molecule pair together with complementary bases on the opposite strand of DNA to form the rungs of the DNA ladder. The bases always pair together in the same way: adenine with thymine, and cytosine with guanine. Each pair is joined together by hydrogen bonds. DNA composed of a sugar phosphate backbone and nitrogenous bases. As you notice, each strand of DNA has beginning and an end, called 5' and 3'. The two strands run in the opposite direction to each other, so that one runs 5' to 3' direction, and one runs to 3' to 5' direction. They are called the sense strand and the anti-sense strand. The 5' to 3' designations refer to the number of carbon atom in deoxyribose sugar molecule to which a phosphate group bonds. This diagram is a detailed process on how DNA replication works. We all know that DNA replication is the process by which DNA makes a copy of itself during cell division. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks hydrogen bonds holding the complementary bases of DNA together, wherein adenine with thymine, cytosine with guanine. The separation of the two single strands of DNA creates Y-shape called a replication fork. The two separated strands will act as a templates for making the new strands of DNA. One of the strands is oriented in the 3' to 5' direction towards the replication fork. This is the leading strand. The other strand is oriented into 5' to 3' direction away from the replication fork. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. Another diagram for DNA replication, wherein you can see the helicase. This helicase is an enzyme binds to the DNA and starts to unzip two strands by breaking the hydrogen bonds. You can also notice in this diagram the separation of two single strands of DNA. That creates what we call replication fork. It is a Y shape, providing template for creating a new strand. The enzyme called RNA primase makes a short strand of RNA primer that is complementary to the template DNA strand. RNA primase also starts to bind at the DNA template.
Next, leading strand formation. In leading strand formation, we have here the RNA primase in the replication fork starts to make bases of RNA called the RNA primer. When we say RNA primer, is that about 5 to 10 nucleotides long complementary to the DNA. RNA primers also are used by living organisms in the initiation of synthesizing a strand of DNA. The example of primers being used to enable DNA synthesis is reverse transcription. When we say reverse transcription, is an enzyme that uses a template strand of RNA to synthesize a complementary strand of DNA. The primer with 3-OH or the hydroxyl and starts the construction of new strand DNA. When we say primase, is an enzyme that synthesize short RNA sequence called primers. These primers serve as a starting point of DNA synthesis since primase produce RNA molecules. That enzyme is a type of RNA polymerase. So, an enzyme called DNA polymerase 3 binds to the end of 3-OH or the 3-hydroxyl end of RNA primer and will make the new strand of RNA or leading strand continuously toward the replication fork. So when we say RNA primer in the DNA polymerase is a short stretch of nucleic acid made up of the single-stranded RNA molecule. An RNA polymerase, called it DNA primase, synthesize a short stretch of single-stranded RNA molecule for starting replication. It is very essentially required for a DNA polymerase to start its catalytic activity. DNA polymerase 3 bind to the primers on the leading and lagging strands and synthesize new DNA from the 3 hydroxyls. DNA polymerase cannot synthesize new DNA without a pre-existing 3-OH or 3-hydroxyl. Thus, DNA replication requires an RNA primer to initiate strand formation. DNA polymerase 3 can only add DNA bases in one direction from the 5' and 5 to the 3' and 3. DNA polymerase 3 is reading the 3 to 5 strand of the old template and creating the new DNA in the 5 to 3 direction. This means it is adding nucleotides to the 3-hydroxyl end of the new DNA strand being made. So what does it mean? The DNA is always synthesized in the 5 to 3 direction, meaning that nucleotides are added only to the 3 end of the growing strand. The 5-phosphate group of the new nucleotides bind to the 3-OH group of the last nucleotides of the growing strand. One of new strand of DNA, the leading strand is made continuously. The DNA polymerase III adding bases one by one in the 5 end to the 3 end direction. When the DNA polymerase III adds a new nucleotides to the DNA strand, it forms a covalent phosphodiester between the adjacent nucleotides 3-hydroxyl group and the new 5-phosphate group. Based on this, the leading strand is made continuously because the DNA polymerase is moving in the same direction as the replication fork. The lagging strand cannot be made in 5 end to 3 end because it runs in the opposite direction. So, the DNA polymerase add nucleotides to the deoxyribose 3-ended strand in a 5 to 3 direction. The lagging strand is synthesized in fragments. Nucleotides cannot be added to the phosphate 5 end because DNA polymerase can only add DNA nucleotides in a 5 to 3 direction. The DNA polymerase III can only make the lagging strand in a series of a small section called Okazaki fragments away from the replic fork. When we say DNA polymerase III is a component of the replisomes which is located at the replication fork, lagging strand can only be replicated in small sections due to the 5 to 3 direction dependence of DNA polymerase III. What does it mean to say that extension by DNA polymerase III proceeds 5 to 3? The DNA polymerase attaches to 3 end of an Okazaki fragment as it moves in 5 to 3 direction. It moves the RNA primer ahead of it and replaces the ribonucleotides with deoxyribonucleotides. Okazaki fragment is started with an RNA primer made by primase since this has 3 hydroxyl end. Okazaki fragments are a small section of DNA that are formed during this continuous synthesis of the lagging strand during DNA replication. DNA polymerase III binds to the 3-hydroxyl end of every RNA primer to add a short row of DNA bases in the 5 end to 3 end direction to make Okazaki fragment. The primase generate short strand of RNA that binds to the single-stranded DNA to initiate DNA synthesis by the DNA polymerase. This enzyme can work only in the 5 to 3 direction, so it replicates the leading strand continuously. Next, let's move to the replication to new DNAs. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. When we say exonuclease are enzymes that work by cleaving nucleotides one at a time from the end of polynucleotides chain. A hydrolyzing reaction that breaks phosphodiester bonds at either the 3 or the 5 end occurs. The removal of RNA primers left a gap, but another DNA polymerase I enzyme then fills D and PTS in the gaps that are left behind DNA. The DNA polymerase I is recognized as an enzyme that fills any remaining gaps between Okazaki fragments and removes RNA primers with a duplex-specific 5 to 3 exonuclease activity, providing a nick that can be sealed by DNA ligase. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand with a phosphodiester band. The DNA ligase play an essential role in maintaining genomic integrity by joining breaks in the phosphodiester backbone of DNA that occur during replication and recombination and as a consequence of DNA damage and its repair. Next, genome structure have single circular chromosome in bacteria, while in eukaryotes have multiple linear chromosomes. The number of origins per chromosome is single in bacteria, while in eukaryotes is multiple. Rate of replication 1000 nucleotides per seconds in bacteria, 100 nucleotides per second in eukaryotes. Telomerase is not present in bacteria, but in eukaryotes is present. RNA primer removal is DNA pol 1 in bacteria, but RNase H in eukaryotes. Strand elongation is DNA polymerase 3 in bacteria, then polymerase delta and polymerase epsilon in eukaryotes.
Transcription. Transcription is enzymatically similar to DNA replication. Both process of replication and transcription involves enzyme that synthesize new strand of nucleic acid, complementary to DNA strand. However, the major difference is that DNA polymerase adds the oxyribonucleic acid on the template of the DNA, while the enzyme RNA polymerase carries out transcription and ribonucleotides on the template of DNA. It is divided into three processes. These are the initiation, elongation, and termination. Transcription is the process by which information in a strand of DNA is copied into a new molecule, mRNA. Why transcription is similar to DNA replication? It is similar because they are both involved in the generation of a new copy of the DNA in a cell. DNA replication makes another copy of DNA, and the DNA transcription it involves in replicating the DNA into RNA. Site for RNA synthesis. Next is beta, with the symbol of B. It is the second largest subunit involved in RNA synthesis. Next is alphas, third largest unit. Under alphas, there is alpha 1 that it has a domain N, or N terminal domain has a role in assembly of RNA polymerase enzyme. Then alpha 2 has a domain C terminal domain has a role in interaction with the promoter, a non-specific binding to the DNA sequence. And the last unit is the omega. It is the smallest subunit and facilitates the RNA polymerase and stabilize the RNA polymerase. Sigma factor for initiation of transcription. RNA polymerase cannot start initiation of transcription by its own. It requires the help of a factor known as the sigma factor. In order for the RNA polymerase to start initiation of transcription, it needed the help of the sigma factor. Sigma factor is a protein that allows specific binding of RNA polymerase with the promoter. So the complete RNA polymerase holoenzyme has 6 subunits. Sigma factor is a protein needed for initiation of transcription in bacteria. It is a bacterial transcription initiation factor that can enables binding of RNA polymerase to gene promoters. Initiation of transcription in prokaryotes. The initiation is divided into 3 steps. First, closed complex. Second, open complex. Third, abortive initiation. In the first step, the RNA polymerase binds to the promoter of DNA region to template. The DNA at this stage is still in the double-stranded form, and this complex is called the closed complex. The closed complex represents RNA polymerase that has just bound with the promoter. Closed complex is the first stage in the transcription initiation. It is the formation of a complex between holoenzyme and the DNA sequence at the promoter, which is in the form of a double-stranded DNA. The next step, the DNA strands near the transcription start site defined as positive 1 unwind to form an open complex. Once in it is formed, the enzyme starts adding RNTPs in the 5 to 3 direction on the template DNA. It should be noted that unlike DNA polymerase, RNA polymerase does not require a primer for the initiation of transcription. Open complex forms during the transcription stage of protein synthesis after the less stable closed complex. Now, once the short stretches of RNA are formed, the open complex now undergoes to abortive initiation. In abortive initiation, the RNA polymerase forms a short RNA stretches, or it is the oligomeric, about 10 abortive transcripts which are released. It is formed because the sigma factor blocks the RNA exit channel of RNA polymerase enzyme. The sigma factor eventually dissociates from the holoenzyme and elongation proceeds. Abortive initiation is also known as abortive transcription, it is an early process of genetic transcription in which RNA polymerase binds to a DNA promoter, and it enters into cycles of synthesis of short mRNA transcripts, which are released before the transcription complex and it leaves the promoters. For bacterial RNA polymerase, the release of the sigma factor marks the end of the initiation process. The sigma factor is released when transcripts are between 9 and 16 nucleotides long, and the length depends on the promoter and the early transcribed sequence.
So I am Paul J. Katipag, assigned to discuss the elongation of transcription, one of the process of transcription of central dogma in prokaryotes. So basically this elongation is making the RNA transcript longer. So yeah, once the sigma factor is released, the RNA polymerase proceeds for the process of the process elongation. So during this process, RNA polymerase walks along one strand of DNA known as the template strand in the 3' to 5' direction. So the RNA synthesis is like the DNA replication, that is always 5', that is always from 5' to 3' direction. So the RNA polymerase is also always moves from 5' to 3' direction. So this directionally has advantage or biological advantage, such as efficient DNA repair and 3' covalent extension of nucleic acid synthesis. So this rule is same on both the strands, whether it is single-stranded or double-stranded. The two strands in DNA are antiparallel and complementary. Therefore, same directionally is maintained during transcription. So for each nucleotide in the template, RNA polymerase adds a matching complementary RNA nucleotide to the 3' end of the RNA strand. So yeah, the RNA transcript is nearly identical to the non-template or the coding strand of DNA. However, these RNA strands have the base uracil, as we were taught by Miss Ma'am, in place of thymine, as well as a slightly different sugar in the nucleotide. So as we can see in the diagram above, if we zoom it in, HT or the thymine of coding strand is replaced with an uracil in the RNA transcript. So the picture presented below shows the DNA being transcribed by many RNA polymerases at the same time, each with an RNA tail trailing behind it. The polymerases near the start of gene have a short RNA tail or have short RNA tails, which get longer and longer as the polymerase transcribes more of the gene. So once the RNA polymerase has transcribed the gene, it must stop to release the RNA product. So how do we release the RNA product? There are two modes of termination in prokaryotes, known as the RHO-dependent and RHO-independent. So RHO-dependent termination depends basically on the protein known as the RHO factor. What is this RHO factor? This is a adenosine triphosphate dependent hexameric helicase. It binds to this sequence and starts climbing up the transcript toward RNA polymerase. So the RHO, the RHO protein binds to the single-stranded RNA rich in cytosine. These cytosine residues are also called the RHO utilization site or the RUT site. Please zoom in to see the yellow 6 thing on the image. Now, when the RNA polymerase reaches hundreds of nucleotides away from the RUT site, it stops the transcription, as you can see at the image. Now, the sequence that holds the RNA polymerase are known as RHO-sensitive post site. Once the RHO proteins binds to the RNA, it uses energy of adenosine triphosphate and translocate at the DNA-RNA hybrid and unwinds the region. Now, while the RHO independent termination does not require any external proteins or factors or the RHO factors for the termination to occur. The RHO independent termination is also called the intrinsic termination, which consists of two sequence elements. It is the short inverted repeats, that is about 20 nucleotides, and stretch of 8 adenosine and adenine and thymine base pair. So once the RNA polymerase has transcribed the inverted repeat sequence, please zoom in the image. The resulting RNA forms a stem loop structure which resembles a hairpin by base pairing itself. So the next step after making the hairpin structure halts the RNA polymerase enzymes or stops the RNA polymerase enzyme. Now, the inverted repeat sequence is further followed by ATR-rich sequence. So the ATR-rich sequence after transcription forms an AU base pair or the adenine uracil base pair, which are the weakest of all base pair, even weaker than adenine and thymine base pair. And because of this, the RNA finally released, ending the transcription.
The difference between prokaryotic transcription and eukaryotic transcription. In prokaryotic transcription, transcription and translation occur simultaneously. Prokaryotic transcription occurs in the cytoplasm, and RNAs are released and processed in the cytoplasm under prokaryotic transcription. Also, under this, RNA polymerases are a complex of 5 polypeptides, and it doesn't require any proteins or other factors for the initiation of transcription. While in eukaryotic transcription, transcription and translation don't occur simultaneously. Eukaryotic transcription occurs in the nucleus and translation occurs in the cytoplasm. And when it comes to RNA, RNAs are released and processed in the nucleus in eukaryotic transcription. Also under this, RNA polymerases are a complex of 10 to 15 polypeptides, and it requires proteins known as transcription factors for the initiation. So let's define what is prokaryotic transcription to be more clarified. So what is prokaryotic transcription? It is a transcription in which often involves several genes, resulting in polycistronic mRNAs that specify multiple proteins in a single molecule. Multiple transcriptions and translation processes occurring in or at the same time in the same DNA template may quickly raise a bacterial protein's intracellular level. The prokaryotic transcription proceeds in 3 steps: initiation, elongation, and termination. And this process is driven by DNA-dependent RNA polymerase enzyme, which transcribes the DNA. Next is, what is eukaryotic transcription? Eukaryotic transcription is the transcription of eukaryotes is much more complicated than that of prokaryotes. In contrast to bacterial RNA polymerase, it can form a connection with the DNA template on its own. Eukaryotes need several additional proteins known as transcription factors to first bind to the promoter region and then aid in the selection of the proper polymerase. So let's proceed to the translation. Translation involves translating the sequence of a messenger RNA or mRNA molecule to a sequence of amino acids during protein synthesis. It is the process in which ribosomes in the cytoplasm or synthesize proteins after the process of transcription of DNA to RNA. Translation undergoes three processes, which is the initiation, elongation, and termination. So, in translation, we can use the word to carry across in relation to translation, or we can use to carry across to easily define translation. In this case, what is being carried across is information that originally was in the genome, enshrined in DNA, then gets transcribed into messenger RNA or the mRNA. And then, that information is translated from the messenger RNA to a protein. So we're taking the same information, but it's going from one form to another, a nucleic acid code to an amino acid code in a protein. The translation is done not in individual letters. It's very much like the human language or any other language that, in this case, all the words are the same length. They're all 3 words long, and the reader in this case is sometimes called a ribosome, which is this big multi-subunit molecular machine that travels along the mRNA. And it reads much like a person reading Braille does. It reads along, detects what are these letters underneath it, and when it detects what those 3 letters are, it decides what the amino acid is supposed to be that it adds to the growing amino acid chain, polypeptide chain, to become a protein. Those mRNA letters are called a codon, and each codon codes for a different amino acid, and eventually those amino acids are all joined together to assemble a protein. Now, let's proceed to the initiation of translation in prokaryotes. The small number 1, or the first step, is the small ribosomal subunit is separated from the large subunit with the help of 2 initiation factors, which is the IF1 and IF3. So we can see here in the picture is the ribosome. So the small ribosomal subunit, or the you can see in the picture the 30S, will be separated with the help of the initiation factors, which is the IF1 and the IF3. The next process will be this complex then binds to the purine-rich region, the Shine-Dalgarno sequence, upstream of the AUG start codon on the mRNA. Now, we can see in this next picture is that the small ribosomal subunit is now separated with the help of the IF1 and IF3 initiation factors, and then move into the Shine-Dalgarno sequence. Next is number 3. The Shine-Dalgarno sequence is base paired to a complementary sequence on the 16S rRNA, which is a component of the small subunit. This alignment ensures that the start codon is in the right position within the ribosome. So the small ribosome is now set in the sequence on the 16SR in the mRNA. As you can see here, the AUG sequence. Now that the small ribosome it will get the 16S rRNA. The next process will be another initiation factor, IF2, brings in the initiator tRNA, which is the cross-like cross-like picture cross-like picture on the right side, which is the separated one. And now with the help of the IF2, it brings the initiator, which is the tRNA, as you can see in here in the fourth picture, charged with the initiator, amino acid, and formyl metathione. So the initiator or the tRNA is charged with the N-formyl metathione. Now the last is the large ribosomal subunit joins the complex and all initiation factors are released. So now that the two ribosomes, the bigger one and the small ribosomal subunit one, they will have a complex and big ribosomal unit or the large ribosomal unit, as you can see in this picture, will now join the complex and all initiation factors are released. So, after being joined, the initiation factors will now then be released. So, in the fifth picture, which is the final picture of the initiation of translation in prokaryotes, we will see it in a bigger picture. So, here, the ribosome has three sites. The A site is the entry site for new tRNA, charged with amino acid or amino acyl tRNA. So it is said here that the new site for the tRNA is the A site, which is in the right side of the P, which is charged with amino acid or amino acyl tRNA. Next one is the P site. The P site is occupied by peptidyl tRNA, which is the tRNA that carries the growing polypeptide chain. So, on the middle part, which is the P site, it is occupied by a peptidyl tRNA. So, this peptidyl tRNA carries the growing polypeptide chain. As you can see here, the P is occupied by this red figure. Now, that in the E site, which is the left side of the P, it is the site in which it is the exit site for the tRNA after it's done delivering the amino acid. The initiator tRNA is positioned in the P site. So, with the explanation in here is that the red figure, which is the peptidyl tRNA, which carries the growing polypeptide chain, it delivers the amino acid. Now that the first is the entry site, which will the peptidyl tRNA will enter and then position in the letter P. Now, when the figure or the peptidyl tRNA is positioned, it will deliver the amino acids required and then will exit on the E site. So the process will be from right to left process. So let's proceed to the another process of translation which is the elongation of translation in prokaryotes. Now, the first step or the first one is a new tRNA carrying an amino acid enters the A site of the ribosome. So, let's go back to the last picture we have visited, which is the the ribosomes are attached to the mRNA, and then there is an attached polypeptidyl tRNA on the P site. Now, in this elongation of translation in prokaryotes, there is a new tRNA there's a new tRNA in this picture, and of course, as we have said earlier, letter A or the A site is the entrance for the new tRNA to come. So the second part is on the ribosomes. In the bigger or in the large ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. So as you can see in the picture, that the new tRNA, which is now attached to the A site, has match against the mRNA codon positioned under the A site. So we can see in the picture that in the mRNA, it consists of triple U, and now that the new tRNA enters the A site, it matches the anticodon of the new tRNA is matched in the mRNA's codon. Now that the new tRNA comes, during this proofreading, tRNA with correct anticodons are rejected and replaced by new tRNA that are again changed immediately. So as we can see in the third picture, there is a proofreading in which there will be a recorrection of the anticodons. As you can see here, the new tRNA is now being replaced by applicable or appropriate anticodons. Now that it has replaced in the fourth one, when the right amino acyl tRNA enters the A site, a peptide bond is made between the two new adjacent amino acid. Now that the new tRNA or the amino acyl tRNA enters the A site and has the right or appropriate anticodons, it will now then attach to the A site between the two now adjacent amino acids. So the new tRNA is replaced with appropriate and now is attached on the A site. So the continuation in this elongation of translation in prokaryotes, last time or the last picture we have seen is that when the right amino acyl tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. Which it says on the last picture is that the P site and the A site is occupied by two tRNA. Now, as the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward in the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. So as you can see in the picture, the E site and the P site is occupied and the A site now is empty. So the question is, how is it empty? For in the last picture, only the P site and the A site is occupied. So according to the information stated above, the tRNA in the P site releases the amino acids on the tRNA on the A site and becomes empty. So it means that the tRNA that is in the P site before, which is the, or we can call it the peptidyl tRNA, releases amino acids. Now that this tRNA belongs to the P site before, it releases amino acids to the ones or to the tRNA that is attached on the A site before. Now that it releases amino acids, it means that it is now empty. Now that it is empty, the ribosomes moves one triplet forward on the mRNA. One triplet forward means that they will move 3 times. So yes, they will move 3 times forward. Now that they will move 3 times forward, the tRNA that is attached on the P before will now go to the E site, and the ones who have received the amino acids will proceed to the P site. So the cycle is repeated for each codon on the mRNA. So as we can see here in the picture, the process is continuous. If one goes out, then one comes in and delivers amino acids to the there's a deliveration of amino acids to each tRNA. Okay, from the word elongation, we can see here in the picture that the process continues, and we can see that it is moving forward and forward. From the word elongation, there is a sequence of increasing the length of the mRNA. So now let's proceed to the last process, which is called the termination of translation in prokaryotes. So in the last part, which is the elongation, the cycle is repeated for each codon on the mRNA. So from the word elongation, this is the process in which it increases the length of the sequence of the mRNA. So now let's proceed to the termination. It happens when one of the 3 stop codons is positioned in the A site. So now in this part, there is 3 stop codons that is positioned on the A site, or the entrance to the ribosome. So now there is a 3 stop codon, which means that this 3 stop codon, it has no tRNA. There is no tRNA that can fit in the A site at that point, as there are no tRNA that match the sequence. So the 3 stop codons, even though there are tRNAs, yet such tRNA do not match to the sequence in the mRNA. So now, at this point, these codons are now recognized by a protein, a release factor. So instead of tRNA, it will be occupied by a protein. So this protein will be a release factors. Now on the last part of the termination, now that the protein is now that the codons are recognized by the protein, there is a binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. So the protein will catalyzes the cleavage of the bond, which will cause the breakup or the from the word termination, the process is now terminated. Now, the polypeptide is released from the ribosome. So as you can see here, that the polypeptides are now released. The ribosome is disassociated into subunits and is ready for a new round of translation. So as we can see in the third picture that there is a termination of the process and the the ribosome is disassociated or separated into subunits. And now it is ready again for another set or another round of translation. So there's a note here that the newly made polypeptide usually requires additional modifications and folding before it can become an active protein. So, to sum up the prokaryotic translation and eukaryotic translation, the definition of prokaryotic translation is that the translation and transcription process is synchronous. And now, the eukaryotic translation is that the translation and transcription process is discontinuous. When it comes to mRNA, in prokaryotic translation, it happens in the cytoplasm. And in the eukaryotic translation, it happens in the nucleus. When it comes to cap initiation, prokaryotic is cap independent or not dependent. And in the eukaryotic translation, it is a cap dependent and cap independent. So we can consider eukaryotic translation as multitasking. It can be dependent or independent when it comes to cap initiation. So when it comes to performance or ribosomes performance or how many ribosomes performed, I should say, in prokaryotic translation, there are 70S ribosomes, and in eukaryotic translation, there are 80S ribosomes. When it comes to stability of mRNA, eukaryotic translation is more stable than the prokaryotic translation. When it comes to ribosomes, in prokaryotic translation, 30S and 50S is equals to 70S, and in the eukaryotic translation, it is 40S and 60S equals to 80S. The lifespan of mRNA in prokaryotic is a few seconds to 2 minutes. When it comes to eukaryotic translation, a few hours to days. So we can see in this description that eukaryotic translation has more lifespan than of prokaryotic translation. So in the occurrence, in prokaryotic translation, there is no definite phase, while in the eukaryotic translation, the occurrence is in the G1 and the G2 phase of the cell cycle. When it comes to the speed of their processes, eukaryotic translation is slower than of prokaryotic translation. So when it comes to their release factors, prokaryotic is in the RF1 and RF2, and the eukaryotic translation is the ERF. When it comes to the numbers of initiation factors, prokaryotic consists of 3 initiation factors and eukaryotic consists of 9 initiation factors. So that would be all for the translation of prokaryotes and eukaryotes. Thank you for listening and God bless to everyone.